Hello viewers, today we will be going to discuss on some very important aspects of lipid metabolism. We will start off with the details of the oxidation of unsaturated fatty acids. Unsaturated fatty acids are predominant in the triglycerides and the phospholipids present in plants and animals. Now, what is an unsaturated fatty acid? It is a fatty acid in which there is at least one double bond present in the carbon skeleton. These are of two types: monounsaturated fatty acid or MUFA, in which there is a single double bond present in the carbon skeleton, and the second one is the polyunsaturated fatty acid or PUFA, in which there are more than one double bonds present in the carbon skeleton. Now, in this particular slide, an example of MUFA is shown. Its name is oleic acid. Its systematic name is cis-delta-9 octadecenoic acid. That means there is a single double bond present between the carbon atoms 9 and 10. This is an example of a polyunsaturated fatty acid or PUFA. It is called linoleic acid and the systematic name is cis-delta-9,12 octadeca dienoic acid. This means that it has got two double bonds present between carbon atoms 9 and 10 and between the carbon atoms 12 and 13. Now, the double bonds present in the fatty acids, that is the unsaturated ones, pose two different problems during their beta oxidation pathway. First, problem number 1. This is the presence of a beta-gamma or a 3,4 double bond. Now, if a beta-gamma or a 3,4 double bond is present, and that too in the cis position, then that is not a very proper substrate for the usual beta oxidation pathway, that is the enoyl-CoA hydratase enzyme. So, it has to be changed into a trans delta 2 double bond, that is a double bond present between the carbon atoms 2 and 3 with the help of an enzyme which is called enoyl-CoA isomerase. Now, this slide actually will show you how the enoyl-CoA isomerase enzyme works. It acts on the trans delta 3 enoyl-CoA or a cis delta 3 enoyl-CoA and converts both of them to trans delta 2 enoyl-CoA, and this trans delta 2 enoyl-CoA is the proper substrate of the enzyme enoyl-CoA hydratase. So, this is how the problem is solved in case of the oxidation of monounsaturated fatty acids. In this slide, we will see the diagram of the beta oxidation of oleoyl-CoA, which contains a single double bond between the carbon atoms 9 and 10. As is shown, the 3 cycles of beta oxidation proceed uninterrupted, releasing 3 molecules of acetyl-CoA. And then the problem arises after the third round. That is, there is a formation of a cis delta 3 enoyl-CoA, which is a poor substrate for the enzyme enoyl-CoA hydratase. So, the enzyme enoyl-CoA isomerase converts the cis delta 3 double bonded enoyl-CoA to a trans delta 2 double bonded enoyl-CoA, and then the usual pathway of the beta oxidation proceeds. So, after 5 cycles, 6 molecules of acetyl-CoA are formed and the beta oxidation of oleoyl-CoA gets completed. Now, coming to problem number 2. This is the presence of a gamma delta or a 4,5 double bond. This problem arises in the 5th round of beta oxidation pathway. Now, presence of double bond between the 4th and the 5th carbon atoms is a result of the product which is called the trans delta 2 cis delta 4 enoyl-CoA. So, what happens? 2 different enzymes come and solve this problem. The first one is called the enoyl-CoA isomerase and the second one is called the 2,4-dienoyl-CoA reductase. Coming to the action of the enzyme 2,4-dienoyl-CoA reductase, this is a NADPH dependent enzyme. It utilizes NADPH for its oxidation and it actually converts the double bond present in a trans delta 2 cis delta 4 enoyl-CoA to a trans delta 2 enoyl-CoA, which is a regular substrate of the enoyl-CoA hydratase enzyme. In this slide, we will see the beta oxidation of linoleoyl-CoA. Now, in case of linoleoyl-CoA, there are two double bonds present between the carbon atoms 9 and 10 and between the carbon atoms 12 and 13. Now, the enzyme enoyl-CoA isomerase actually converts the double bond present between the carbon atoms 3 and 4 into the carbon atoms 2 and 3. But the double bond between 4 and 5 still exists. So, how to solve this scenario? The enzyme 2,4-dienoyl-CoA reductase then comes and they convert the double bond present in between carbon atoms 4 and 5 to the double bond present in between the carbon atoms 3 and 4. Now the enzyme enoyl-CoA isomerase once more comes and converts the double bond between the carbon atoms 3 and 4 to the double bond between the carbon atoms 2 and 3, so that the problem is now solved with the help of these two auxiliary enzymes, the enoyl-CoA isomerase and the 2,4-dienoyl-CoA reductase, and the normal beta oxidation pathway may continue as usual. Coming to the ATP yield in case of the beta oxidation of unsaturated fatty acids, the ATP yield or the energy cost is only very less in comparison to the beta oxidation of the saturated fatty acid. Why this is so? Because there are fewer hydrogen atoms present in the unsaturated fatty acids. So, a fewer number of electrons will enter the respiratory chain to get oxidized and release metabolic energy. This is why the unsaturated fatty acids beta oxidation releases lesser amount of energy than the beta oxidation of the saturated fatty acids. Odd chain fatty acids are not very common in nature. However, they may be present in plants and marine organisms. Some amount of the 3-carbon propionate is added to bread as mold inhibitors, and through this route they may enter our system. 3-carbon propionate may also be arisen out of fermentative metabolism within the digestive tract of cattle and similar other ruminant animals. Also, the 3-carbon propionate is generated out of the metabolism between the branched chain amino acids like the isoleucine, valine and methionine. Now, coming to the oxidation of odd chain fatty acids in proper, at the last step of the beta oxidation, instead of 2 acetyl-CoA molecules which are produced, here there is the formation of a 5-carbon compound. This 5-carbon compound is splitted up to release 2 compounds. One is the 3-carbon propionyl-CoA and the next one is the 2-carbon acetyl-CoA. Now what happens to this propionyl-CoA? This propionyl-CoA actually cannot enter the TCA cycle on its own. So, it has to be converted either to succinyl-CoA by the methyl malonate pathway or to acetyl-CoA by the beta hydroxy propionate pathway. So, firstly, the methyl malonate pathway. In this pathway, there are 4 steps. In the first step, propionyl-CoA gets converted to D-methyl malonyl-CoA. then this D-methylmalonyl-CoA is racemized to form L-methylmalonyl-CoA, and after that this L-methylmalonyl-CoA forms Succinyl-CoA. So coming to the stepwise discussion of methyl malonate pathway, as this slide shows, this is step number 1, where propionyl-CoA gets converted to D-methylmalonyl-CoA with the help of the biotin-dependent enzyme propionyl-CoA carboxylase. Coming to step number 2, the D-methylmalonyl-CoA is converted to L-methylmalonyl-CoA by the enzyme methylmalonyl-CoA racemase. Coming to step number 3, this L-methylmalonyl-CoA will be converted to succinyl-CoA with the help of the enzyme methylmalonyl-CoA mutase. But one interesting thing in this conversion is the participation of what is known as coenzyme B12. This coenzyme B12 is very crucial for this reaction. Now coming to coenzyme B12, its systematic name is 5'-deoxyadenosylcobalamin, which means that it is a derivative of the vitamin B12 cyanocobalamin. Its structure is shown in this slide, which is a bit complex one. Now we have to know the role of this coenzyme B12 in the last step of the methyl malonate pathway. In this reaction, we can see that the L-methylmalonyl-CoA is getting converted to succinyl-CoA through an intramolecular rearrangement reaction. That means the acetyl-CoA group, which is originally situated in carbon number 2, is getting shifted to carbon number 3, and the hydrogen atom from carbon number 3 is getting shifted to carbon number 2. So this is a very good example of intramolecular rearrangement. Now coming to a few disorders of propionate metabolism. The first one is called the propionic acidemia. This disorder happens due to the deficiency in the enzyme propionyl-CoA carboxylase. Since this enzyme is non-functional in these patients, there is an accumulation of toxic acids and various other toxins in our brain, heart, and liver that may lead to seizures, as also a delay or an improper walking and talking functions. Coming to the second disorder, it is better known as pernicious anemia. It belongs to the category of anemias which is known as the megaloblastic anemias, and it happens due to a loss of the gastric parietal cells. Now, gastric parietal cells are actually responsible for the release of the intrinsic factor, a protein that is meant for the vitamin B12 absorption in the ileum. So since these gastric parietal cells and the intrinsic factor are absent, then vitamin B12 cannot be absorbed in the ileum, leading to a much decreased number of RBCs and a resultant anemic situation. Coming to the third disorder, it is called the methylmalonic acidemia. This happens due to a deficiency of vitamin B12 in the patient. Now, the succinyl-CoA that is generated in the methyl malonate pathway, we have to remember that it cannot enter the TCA cycle directly, but it has to be first converted to succinate, and then to fumarate, then to malate, and then to pyruvate once more. So coming to this scheme, succinyl-CoA first gets converted to succinate, then this succinate gets converted to fumarate and malate, and once this malate reaches a good concentration in the mitochondrial matrix, it comes out into the cytosol and gets acted upon by the malic enzyme, which is also known as malate dehydrogenase decarboxylating, when the malate suffers an oxidative decarboxylation to yield pyruvate with the release of carbon dioxide. Now this pyruvate will get converted to acetyl-CoA, and the acetyl-CoA will enter the TCA cycle to yield metabolic energy. Now the methyl malonate pathway actually takes place in those organisms in which there is coenzyme B12. But in case of those organisms in which coenzyme B12 is not present, for example in case of plants, we have the beta hydroxy propionate pathway, in which the propionyl-CoA generated out of the beta oxidation of the OR chain fatty acids get directly converted to acetyl-CoA, and this acetyl-CoA then enters the TCA cycle and gets further oxidized. Now, another interesting aspect of lipid metabolism are the ketone bodies. The ketone bodies are of three different types: the acetoacetate, the acetone, and the D3-hydroxybutyrate. Now, what is ketogenesis? Ketogenesis is defined as the synthesis of ketone bodies in the liver and kidney mitochondria. Now, we have to learn when ketogenesis takes place. When the carbohydrate metabolism and the fatty acid metabolism are perfectly balanced, then ketogenesis doesn't take place. It happens only in case of a poor carbohydrate metabolic pathway. Coming to the reasons for ketogenesis, if the carbohydrate molecules are not properly utilized, or if they are not available to us, then there will be hardly any synthesis of oxaloacetate by the TCA cycle. But side by side the fatty acid oxidation is continuing, so acetyl-CoA molecules are generated as usual. So what will happen to this acetyl-CoA? It cannot condense with oxaloacetate because it is not properly produced. So the acetyl-CoA will be accumulated within our body. The second condition is the fasting, starvation, or untreated diabetes mellitus conditions, in which the oxaloacetate, however minimum it is produced, is diverted to generate glucose by the process of gluconeogenesis. So acetyl-CoA is left alone once more. It has got nobody to condense with, so the acetyl-CoA gets accumulated and gets diverted to the pathway called ketogenesis and forms ketone bodies in the liver and kidney mitochondria. Now, as we can see in this slide, there are 4 steps to ketogenesis. In step number 1, 2 molecules of acetyl-CoA condense with the help of the enzyme thiolase to form acetoacetyl-CoA. This acetoacetyl-CoA will then undergo reaction with acetyl-CoA with the help of the enzyme hydroxymethyl-glutaryl-CoA synthase or HMG-CoA synthase to yield hydroxymethyl-glutaryl-CoA. This hydroxymethyl-glutaryl-CoA or HMG-CoA is then lysed by the help of the HMG-CoA lyase to yield acetoacetate, the first ketone body in this pathway. This acetoacetate has got 2 alternative fates. It may suffer a non-enzymatic and as a result a slow and spontaneous decarboxylation to yield the second ketone body, which is the acetone. In the second alternative pathway, acetoacetate may suffer a reduction at its carbon atom number 2 with the help of the enzyme beta-hydroxybutyrate dehydrogenase to form D3-hydroxybutyrate, the third ketone body in our body. So this particular slide has summed up the 4 steps of ketogenesis. Now coming to a very interesting aspect, sweet odor in breath. In case of untreated diabetes mellitus patients, there is an excessive accumulation of ketone bodies, particularly acetone in their blood. Now, acetone is a compound that has got a sweet odor. So, the breath of the untreated diabetic patients will give out a sweet smell that helps the doctors to recognize that they has got an untreated diabetes mellitus disorder and they must be treated very soon. Now, what is the utility of ketone bodies in our system? The ketone bodies will serve as metabolic fuels. After their synthesis in the liver and the kidney mitochondria, the ketone bodies diffuse out of these two organs into the bloodstream. And through bloodstream, they are carried to the extrahepatic tissues like the cardiac tissues, the skeletal muscle tissues, the renal cortex, and the adipose tissue. In these extrahepatic tissues, the ketone bodies get converted to acetyl-CoA, and then this acetyl-CoA will enter the TCA cycle to generate a lot of metabolic energy. This slide will show you how the ketone bodies are getting converted to acetyl-CoA. Firstly, there is the participation of the enzyme D-beta-hydroxybutyrate dehydrogenase. It is a NAD+ dependent enzyme. Using this enzyme, the compound D3-hydroxybutyrate gets converted to acetoacetate. This acetoacetate then reacts with succinyl-CoA and it forms acetoacetyl-CoA. This acetoacetyl-CoA then undergoes a thiolysis reaction to form two molecules of acetyl-CoA with the help of the enzyme thiolase once more. So this pathway will show you how the ketone bodies which are generated out of the improper carbohydrate metabolism gets used up as alternative metabolic fuels in our system. They form acetyl-CoA molecules that enter the TCA cycle to liberate metabolic energy. Now, coming to an abnormal situation even more, the overproduction of ketone bodies. The normal concentration of ketone bodies in our blood is only 1 milligram per deciliter. So when the concentration of the ketone body exceeds this value, we call it an abnormal situation and an overproduction of ketone bodies. This particularly happens in case of three situations: severe starvation, fasting, and untreated diabetes mellitus. The most important overproduction scenario is referred to as the ketosis. Ketosis term includes two medical conditions. The first one is called the ketonemia, that is the overproduction of ketone bodies in our blood. The second situation is known as the ketonuria, that is the overproduction and accumulation of ketone bodies in our urine. And ketonuria follows ketonemia. This chart will show you the normal and the abnormal concentrations of the ketone bodies that are secreted through our urine in milligrams in 24 hours. In case of normal individuals, the value is less than or equal to 125 mg per day. Whereas in case of extreme ketosis, the value increases to about 5000 milligram in 24 hours. Likewise, the blood concentration of the ketone bodies also increases from less than 3 mg per 100 ml of blood to 90 mg per 100 ml of blood in case of untreated diabetes mellitus patients. Coming to the second abnormal situation, which is known as acidosis. Acidosis refers to a lowering of the blood pH due to formation of the ketone bodies D3-hydroxybutyrate and acetoacetate. Now please remember that these two organic acids are very strong ones, and the pKa value of their carboxylic group is around 4. So due to this high pKa value, they can release a lot of protons in our bloodstream, and it may lead to the acidification of the blood. Now, extreme cases of acidosis may lead to coma, and if left untreated, it may even lead to death. Now, ketoacidosis. Ketoacidosis is a medical condition that actually imbibes two different medical conditions that I have already discussed, that are ketosis and acidosis collectively taken together. Now, coming to the treatment of the overproduction of ketone bodies, we have to be very sure that in these patients we maintain a proper water and electrolytic balance, and we must also treat them with insulin from external sources that will stimulate glucose uptake by the tissues and inhibition of ketogenesis. Now, coming to the prevention part of the overproduction of ketone bodies, we have to regularly monitor the blood and the urine of untreated diabetes mellitus patients or the people who live on fasting or starvation conditions, and check whether they are suffering from any overproduction abnormalities. We have summarized some important aspects of lipid metabolism in our today's lecture, that is the oxidation of unsaturated fatty acids, the oxidation of odd chain fatty acids, and a detailed account of ketone bodies and ketogenesis. Thank you so much.